half of life is genetics, half of life is the environment. So be aware of your family tree. Please read this book, Codebreaker by Walter Isaacson. It's about Watson and Crick developing the DNA in the genome. It's about the doctors working on messenger RNA, most of whom were women, with a lot of sexism, by the way. And the Nobel Prize going to Jennifer Dudas for messenger RNA and the future of the vaccines. So we're in the gene editing era where we can take out the bad gene for hypertrophic cardiomyopathy, for amyloid, maybe take out the bad gene that makes LDL, thalassemia. Of course, there's some genetic problems with the misuse of the data and the problem with genetic discrimination. There's a test now for amyloid where you can test your patients. How many doctors here are using it? It's a little scary because, you know, when you're 30 years old, do you want to know you have the gene for amyloid? So right now, our feeling is the genome, the DNA helix is the boss. It runs everything. It tells the whole body what to do. But everything you know about genetics is wrong. So this book by Philip Ball, How Life Works, is about the new genetics. Dr. Ball is a doctor of biology. He was the editor of Nature journal for 20 years. President Clinton said, we are learning the language in which God created life, but the genome is not the book of life. Maybe 15% of what happens in your body is controlled by the genome, but many other things are going on. Living systems are not machines. We mine our environment and manipulate it. There are 37 trillion cells in your body. One more time, 37 trillion cells, each of whom acts on its own. So that if it gets cold, if it gets hot, if you get COVID, if you get cancer, your cells on their own do what they're supposed to do most of the time. So life is not wholly encompassed in the gene. The gene makes proteins, but the proteins can fold in any way they want. And what really does most of the work is RNA, not DNA, which regulates the gene network. So the RNA is the computational engine of the cell. Epigenetics, any process affecting the expression of genes that does not involve a change in the sequence of genes. The problem with CRISPR engineering is you can take out one thing. You can change an A for a T or whatever, but it really only works for the monogenetic disorders. Chromatin can affect gene regulation. Your cell is noisy. Your cells decide their fate, electing what to do. It's almost like each of those 47 trillion cells have their own cognition. And of course, their goal is self-preservation. So where are we now? Eric Topol said 20 years after the human genome was sequenced, there's relatively little to show for it in clinical process.